Okay, lecture 3 will talk about high-resolution purification methods of a different kind. Among those, we'll be talking about one particular separation method, that is chromatography. Okay, chromatography method can come in as in paper chromatography, column chromatography, or gas chromatography. So it can come in different medium, but whatever the medium you use, the principle of chromatography should stay the same. So we'll talk about the principle. Okay, so the word chromatography means something like color writing. That's what it means, because chroma means color in Greek, and graphene is writing in Greek as well. So color writing, basically what it is. So how come it's a color writing? If you think about this paper chromatography, if you separate out different color pigments from plant extract, like a plant leaf extract, things like that, you will see different colored pigment separation on a piece of paper. So it's a color writing. So that's the origin of the word. But like I said, you can use different medium besides the paper. So it's basically a separation technique where you can separate out different molecules on a different medium. Okay. So separation technique based on the differential interaction of molecules between a stationary phase, meaning the medium that is staying put. If it's a paper chromatography, that'll be a piece of paper, or if it's a column chromatography, that's going to be a column filled with resin, or if it's a gas chromatography, it's going to be a gas of different kinds. Okay, and then through that stationary phase, there will be a mobile phase that's going to continuously move through. Then mobile phase will carry your mixture. So the mixture will be separated out on a medium. Okay, so typically the mobile phase could be a buffer of different kind that will be hooked up with a pump. So it'll generate a continuous moving through the medium. Okay, so there's a stationary phase and the mobile phase involved in the chromatography methods. So again, stationary phase could be resin column chromatography. It could be made out of different materials like polysaccharide or cellulose or agarose, or synthetic polymers like polyacrylamide or silica. Okay, mobile phase again, it's typically liquid if it's a liquid chromatography, or it could be gas if it's a gas chromatography. Okay. So molecules will move through a stationary phase in a different rate, because molecules will have different type of interaction on the stationary phase. So based on difference in the interaction, they'll be separated out if you start collecting different molecules coming off from your medium. So it will be something like that. So if you're looking at the column chromatography, so here's the column that contains resin. So your mixture of your molecules are introduced through the mobile phase that's moving through your stationary phase, the column. So in this case, there is no pump. It's just a gravity pull that's going to continuously move your buffer through the column. Okay, but based on the difference among these molecules interaction with the medium, the resin, they will have different speed in terms of their traveling, okay, through the medium. Okay, so the molecules will be separated out on a medium continuously. So if you collect those things that are coming out from the column, different fractions meaning in the separated small volumes, okay, you will see different molecules are contained in different fractions. That is basically the separation principle applies here. Okay. Again, if it's done on a piece of paper, we call that a paper chromatography. So filter paper will do fine. So the filter paper is touching with this buffer. So the buffer will travel through this filter paper. Okay. So on that medium, you will place a mixture of pigment or whatever the molecules. Okay. So the molecules will follow with the buffer. They will travel on the piece of paper. But based on the difference on the molecules interaction, again, they'll be separated out on this paper. So that's the paper chromatography. Okay, very simple. Another type of chromatography is called TLC. TLC stands for thin layer chromatography. In this case, the medium is a plate of silica coated glass plate. Okay, it's a silica coated glass plate, and then when you start out your mixture right here, again that plate is touching the solvent of certain kind. Then solvent will travel, okay, from the bottom to the top. Okay, you can dump it on this side. We can also dump it on the other side as well, so you can do the two-dimensional separation. So that's a thin layer of chromatography. So for example, your nucleotide GATC can be separated out using a thin layer chromatography. Okay, another simple kind of chromatography method is a thin layer chromatography. Okay. And then finally, a column chromatography. In this case, of course, the medium is a column resin. Okay, so that's where your molecules will be separated out. Okay, so in terms of operating your chromatography method, there's going to be a sample loading step where your sample will be added to the column, and then there'll be a washing step, and then there'll be elution step. Okay, among those, elution is a time when the molecules that are bound on your column resin will be recovered from the column. Okay, how do you do that? By challenging the binding that's happening between your molecule and the resin with some kind of a solvent that has some special ingredient, like maybe salt or low or high pH. So the interaction that's still happening between your target molecule and the resin is broken up. So that's when you can capture your target molecule. So that's again the term elution, what that means. Okay, remember elution. Okay. So the capacity of column increases with its volume. So the bigger the column volume is, you can capture more target molecule. That's what it means. Okay. So again, this is a column chromatography method. So same, here's a medium. That's the column filled with the resin. So resin should have some kind of special property. You can make special interaction with your target molecule. So when your sample is applied, there's a sample loading step, and then the interaction will start, and you will do a continuous feeding of your mobile phage. Mobile phage will come through the column. So while the mobile phage is moving through the column, your molecules will be separated out. Okay, the molecule that doesn't have strong interaction will come out of your column first. The molecule that has a stronger interaction will stay there much longer time, and then if it doesn't come up with the normal mobile phage, you can change the mobile phage with something with a special ingredient that will challenge the interaction between your target molecule and the resin. That could be a buffer with a high salt, then your molecule comes out. So that's the elution step. Okay, the sample loading, continuous washing, and then elution. So three main steps existing in the chromatography running method. Okay. So liquid chromatography, again, liquid is the mobile phage, the buffer that's running through the column. Okay, so liquid chromatography method can be called as high performance liquid chromatography, in this case HPLC, or FPLC, which means fast protein liquid chromatography. Okay, fast protein liquid chromatography versus high performance liquid chromatography. Okay, normally when you try to purify and capture your active pharmaceutical ingredient, you are using FPLC, fast protein liquid chromatography. Okay, but HPLC, normally it is done at a small scale where you need to identify a molecule of your interest. So HPLC is done a lot in a small scale as a kind of analytical method to identify a target molecule instead of a purification and capture. Purification and capture normally is done through the FPLC instead of HPLC. Okay, so what's the benefit of HPLC? Since you are applying the higher pressure through the column, it's done very fast, and then it can give you better resolution as well. So it runs 10 times faster because you're applying higher pressure, higher performance, and then it'll give you a good separation, higher resolution, HPLC. That's why it is used a lot as a analytical method. Okay, and FPLC, you're trying to purify and capture your target protein molecule. So its main, downstream purification method is normally FPLC. Okay, so this is a makeup of your column chromatography system normally. Okay, so there's going to be a column right here. Okay, so besides the column, you have the mobile phase container, that's a buffer tank, right? So you should have enough mobile phase to run through all of your molecules and capture your molecules. So your target molecule will be eluted and captured here. Probably you will have something like fraction collectors at the end, okay? And then the pump will generate continuous flow of your mobile phase. There's going to be some kind of pump, maybe peristaltic pump or something will be there. And then there may be some filters that will eliminate whatever the particle that may be floating, some contaminants, or air bubbles. So you don't want to have any big air bubbles running through your column, then that's going to damage your column. So that could be a bubble trap. That's another term for this filter. And then pre-filter again, because you don't want to go any impure stuff going through a column, so there may be a couple of filters there. And then sample is injected through this sample injection port or sample injector loop valve. Okay, the sample is injected there, and sample will be loaded to your column. Okay, and then there will be some detection methods, something like UV spectrophotometer. So UV or visible light spectrophotometer is installed in your chromatography system, so you can visualize the reading observance change during your run. So the continuous recording of your spectrophotometry will be monitored on probably screen, computer screen. We call that a chromatogram. Okay, that will be done. Also, there will be another detector that will normally measure the pH change, because sometimes you change your buffer from neutral pH to maybe low pH or high pH when you try to elute your protein. So pH detection may be important. Another important thing is there may be a conductivity measure in your system. So conductivity will measure what? The change of the salt composition in your buffer system. So that will be detected by your the conductivity measure. So there may be about 3 different detection systems attached within your chromatography system. Okay, pH, conductivity, and spectrophotometer. Okay. So that's a simplified diagram, but the real chromatography system we have in the lab, one of them is biologic LP system. It's this one. Okay, again, there's that buffer tank, and your column is probably located right there, and then it's connected with the tubings, and your pump is right here. Okay, it goes through the pump and come and attach to the pump, and attach to the column, and after the column it comes out, and there will be a UV spectrophotometer and the conductivity measure somewhere, and then it goes down to the fraction collector. Okay, where the fractions will be collected, and then the detection will be visualized on your screen monitor, computer screen monitor. So your UV change, absorption change, or your conductivity change will be monitored continuously. That's a chromatogram. Okay, you'll get to see it, and you'll do a lab on this later on. Okay, as I told you, on the detection method, there are 3 important kinds of detection that are attached to your chromatography system. One is the UV spectrophotometer, okay, especially UV280. Remember, UV260 and 280. 260 is used to measure your nucleic acid concentration. Okay, and then 280 is normally a special wavelength. The protein will be detected. Okay, especially the amino acid with aromatic ring structure, like a tyrosine, tryptophan, phenylalanine, they pick up UV280 a lot. So the chromatography system uses UV280 spectrophotometry to monitor the proteins that are coming off from your column. Okay, so UV spectrophotometer, UV280 is used. And then conductivity, again, it measures what? The change of the salt composition in your buffer. Okay, and the pH again measures the hydrogen ion concentration. Okay, so if you look at the chromatogram, something like that, you will see this one has only 2 lines. So it typically measures UV absorption. So these peaks will correlate the special proteins that are coming out from your column. So they are measured. And then this red line here in this chromatogram is the conductivity change. So low conductivity, and it's creeping up, and then it goes up high and it comes down. Okay, so conductivity will go up if you have high salt composition in your buffer. Okay, higher salt here. Okay, and then it comes down if there isn't much salt there. Okay, and this graph, if you look at it, this x-axis shows you column volume. Column volume is how big your volume is. Your column volume is, let's say your column contains 10 liter. So 1 column volume is 10 liter. So if it says 2 column volume, that means what? 20 liter. Okay, so 2 column volume, 10 to 20 column volume. So sample loading is done with about 2 column volume of buffer introduced to the column. And then this is probably washing. Okay, washing using about 10 to 20 column volume of your